Hello, everyone. I'm Dim Bai from MBZ UAI, and it's my great honor to present here in ISMB 2024. Today, I'll present our work, AttentionPert Accuracy Modeling Multiplex Genetic Perturbations with Multi-Scale Effects. Okay, this is our main table of contents. In biomedical research, it is crucial to correctly model how cells react to genetic perturbations. It helps us understand the mechanisms of many diseases, including cancers, genetic disorders, and some infectious diseases. For example, in cancer, certain genetic mutations can lead to uncontrolled cell growth. Understanding these mutations helps in identifying targets for new drugs. It's also essential for developing new drugs and therapies. Especially accurate predictions of perturbational effects are central to personalized medicine, where treatments are tailored to the genetic profile of an individual's disease. In biochemical labs, genetic perturbation is usually done through CRISPR-based Perturb-seq technology, as shown here. This tech first measures gene expressions of many unperturbed cells. Then it perturbs specific genes of many other cells, which will change the expressions of these cells. Finally, it measures the post-perturbation gene expressions. The number of perturbed genes varies from one to many. However, for multi-gene perturbations, there are fewer high-quality samples. This is simply because of the combinatorial explosion of the number of possible perturbations. When the number of perturbed genes is increasing, the number of possible perturbations increases exponentially. For example, if there are k genes, there are only k single gene perturbations, but there are k choose 2 double gene perturbations. And that's k squared, right? OK squared. Therefore, there are fewer high-quality samples for multi-gene perturbations, and so it's very important and also very hard to predict transcriptional effects of these unseen perturbations. A genetic perturbation dataset is usually produced within the same cell type, such as K562, which contain many cells under different perturbations or no perturbation at all. Here we call it a control perturbation condition, as it means no perturbation. To set this problem generally, we first define unperturbed cells as vectors xi here, and also many perturbed expressions for each c as yci here, where each xi and yci are k-dim vectors, assuming we are interested in k genes in this problem. Then the perturbation condition, the genetic perturbation, is a subset of the set of gene indexes. And the goal is to predict expressions for novel conditions C, the subsets C here, based on the known X and Y's. To do this, people first think of simulating the gene regulatory network, GRN, to deduce the perturbational effects indirectly. However, these methods perform not very well, as the simulation of GRN itself is not an easy task and very noisy. Later, people started utilizing deep neural networks, which directly map genes and perturbations into high-dimension spaces and solve it on latent space. Recently, the previous SOTA method, GEARS, in 2023, a graph-enhanced gene mapping model, focuses on only gene perturbations and outperforms other models largely. After GEARS, a lot of single-cell foundation models, similar to large language models, they are first pre-trained on large-scale unlabeled cell datasets, and then use GEARS as a downstream head to try to perform better on this task. Until now, GEARS is still the SOTA method, as it does not require pre-training, and other foundation models still utilize it. As a non-pre-training model, we basically follow the procedure that GEARS established. First, we take into genetic perturbations, such as C equals to ij here, meaning it perturbs I and j gene, feed it into a deep learning model that predicts the effects, and then add these effects to the unperturbed sample to get the predicted post-perturbation expressions. And finally, we apply the loss that mirroring the error between the predicted values and the ground truth, and use gradient descent to minimize the loss and update the model for each epoch. Our main innovation is the perturbational effects predicting AI, that is called AttentionPert. Without changing the input data or the loss function, our model can perform much better than Gears and also other non-pre-training models. Then let's see our method. This is the overall structure of the AttentionPert. Taking into a perturbation condition, for example, C equals to ij here, two encoders map it to high-dimensional vectors. And then we add them together, input them into a gene-specific decoder, and get the predicted post-perturbation shifts. The two encoders are called PERT-weight, which encodes the non-uniform global effects, and PERT-local, which encodes the local disturbance. Here, locality is defined on a bio-knowledge graph, which I'll cover later. This illustration furthermore explains the goals of our encoders. Our model are based on a fundamental assumption. The unperturbed state of each gene can be represented as a vector in a high dim space. When certain genes undergo perturbation, the basal states of all unperturbed genes are altered into perturbed states. Assuming the perturbation condition here in this illustration is ij, which are both local to k but distant to L in a knowledge graph. PERT-local first alter these vectors locally, meaning changing ij k larger and L less. PERT-local also changes them non-linearly through the non-additive bias module to avoid additivity. I'll cover more about that in experimental results. Then PERT-weight first change these vectors in a general way. That means the same delta vector, pi plus pj here for each gene, and then it computes the weighted gene-specific effects, where the weight matrix denoted as W, generated by attention mechanism here, and alter each gene state accordingly as the linear combination of the weight matrix. And finally, of course, these vectors are decoded into scalars and treated as the shifts. By progress, our model considered both the local effects, the global effects, including the general global effects and the non-uniform global effects, while Gears only considers the uniform global effects. That is, Gears model only covers this part and also other models, this red circle part. When we talk about the local and global effects, we defined the locality in a knowledge graph made of genes. Such a gene-gene interaction graph could be of many forms, such as GRN and so on. We choose to follow and improve over the original gene ontology graph that used by Gears. That's all about our method. For more details, welcome to check our paper because the model is really complicated. Yeah. And let's go to the experiments part. First, we need to define different experimental scenarios. So first of all, every perturbation in the test set is never shown in the training set, as our goal is to predict for novel perturbation. For single gene perturbation, it's easy because there is only one generalization class called sin 0/1, where the only perturbed gene is never seen in the training and validation set as a whole. Yeah. For multiple perturbed genes, there are multiple testing scenarios. For example, for two gene perturbations, there are three scenarios: the sin 0/2, sin 1/2, and the sin 2/2 scenarios, which mean different numbers of genes inside a test perturbation that was seen during the training by the model. For experimental results on harder ones, the two gene perturbations, we test the model on the Norman dataset. We mainly compare the MSE and the Pearson data score of the top 20 differentially expressed DE here, those top 20 DE genes for each perturbation. We can see that our model outperforms CPA and Gears largely, especially on the sin 0/2 cases. This is a perturbation-specific comparison between our models, MSE and Gears, on one split. We can see that for all perturbations of sin 0/2 scenario, and for most of the perturbations of sin 1/2 and sin 2/2 scenarios, our model performs better than gears. We also check the performance of different numbers of top DE genes. As the number grows, the performances generally grow better. For each DE number from 20 to 200, our model all performs better. For simpler single gene perturbations, we carried these experiments on the replogo datasets for RPE1 cells and K562 cells. Results show that our model also performs better than the previous method on single gene perturbations. We have also done the ablation study to explain the necessity of both the per local and per weight modules. We can see from the results that first, per loc alone cannot make competitive results at all. And secondly, although per weight alone also makes good performance on the sin 0/2 cases, its performance in sin 1/2 and sin 2/2 are worse than gears. That's why we finally confirmed that per local and per weight combined together can make great results on all three scenarios. Finally, I'd like to introduce an important feature or subtask that Gears authors proposed and our model also does well in. That is to predict the non-additive effects or synergistic effects. In real-life experiments, effects of perturbation on genes IJ not always equals to effects of I alone and of J alone, as introduced by Dr. David. We can see the illustration here. If the AI learns well of two single gene perturbations, I and J alone, how will it deduce the novel perturbation I plus J? The non-additive nature of gene perturbations requires the AI not to simply add them together. Gears avoid the additivity through a cross-gene layer which disturbs the decoded scalars, while in our model, we use the non-additive bias layer which is inside the encoder and operates only the representation vectors in the latent space. Here are some results showing the ability of our model to perform correctly while predicting non-additive effects. This figure shows the changes of expressions of several genes after perturbing the combination FOXA1 and HOXB9, which is a sin 2/2 case in the test set. The gray bars show the true post-perturbation change of the combination. The orange and blue bars show the separate single gene effects, and the green bars show the prediction of the combination made by our model. Remember that this is a sin 2/2 case, yet our model does not simply add these things together. It learned from the training stage. Instead, it tries to predict the non-additive effects as good as possible, although not perfect. As we can see, the green bars are more similar to the gray bars instead of the summation of the orange and blue bars. This table further proves that in general, the NA bias design is necessary in predicting the non-additive effects. These experiments test the model's performance on non-additive genes with or without NA bias. Here, the non-additive genes are a subset of top 200 DE genes that are given a threshold eta and the perturbation C equals to IJ. It's the set of genes where the non-additive effect is larger than eta times the true expression change. That is the summation of two separate single gene effects minus the combination effect is larger than or equal to the eta times the combination effect. For eta ranging from 0.25 to 0.75, model with NA bias all performs better than the model without it. So all these experimental results fully reveals that NA bias is crucial in modeling the non-additivity of multi-gene perturbations. To summarize our work at AttentionPert, we have two major design innovations. First, we fully categorized and modeled the multi-scale effects, the global and local ones of multi-gene perturbations. Secondly, we apply the non-uniform effects, which are the weighted distinct effects of perturbations upon genes in the representation space. And finally, we have three experimental improvements. First, AttentionPert outperforms previous SOTAs largely in general multi-gene perturbations. And secondly, our model accurately predicts the most OOD scenario, that's sin 0/2 cases. And finally, our experiments reveal its ability to predict non-additive effects precisely by utilizing the non-additive bias module. Yeah, that's all for my presentation. Thanks for your attention. Yeah. And we are also recruiting for research and bioinformatics data scientists. Welcome to contact Professor Eric and if you are interested. Thanks.
Okay, thanks. All right, we have time for some questions. Maybe I'll start with one. So it's nice to see that you did these ablation studies. I was also wondering, for the part that you showed on where you used the gene ontology and then there's also this augmented gene ontology.
Yeah.
Was it also something that you left out of the model to see how much it contributes?
Pardon, what do you mean by left out the model to see?
Yes, in your ablation study, was this also something you...
Oh, yeah, in ablation study, yeah, we also tested the graph that if we don't use the gene ontology graph, if we use a random generated graph and so on, it turns out that this gene ontology graph is useful. And the augmentation of the gene ontology graph is still can make slight improvements over the, especially in the per local encoder, because when we talk about locality in the gene ontology graph, we don't want it to be absolute local. We want it to have a slight leakage of informations between the genes. Yeah, that is not limited to its neighbors. That's why we augment the gene ontology graph. And by the way, gene ontology graph is credited to the Gears authors, not us. Yeah, and yeah, we also did that part. I just didn't show that. Yeah, in our ablation study. Yeah.
Yes, I see we have another question there.
Yeah. So what kind of training data set you use? I assume you use the same as the Gales used.
Which
training data set?
What? Which
training data set?
Training data set. Yeah. We use the same 3 data sets that the Gears used actually, and the same way they pre-processed. Yeah, that they provided. Yeah.
Did you kind of play around, try if you reduce, the amount of the training data set to see the performance of your models?
You mean reduce the training data set?
Because not all the organism as a human has all these kind of rich data sets for the perturbation. Like if you want to have more broad application for this kind of model, will have much less data set in other organisms.
Yeah. Actually, yeah, we actually didn't do that on those less maybe lower quality data sets. As I said, that the data set the Gears is using, especially for 2 gene perturbations, the Norman data set, it's already the highest of the highest quality among all the, especially the multi-gene perturbations scenarios, and so maybe for those other organisms and maybe for those lower quality data sets, we didn't do the experiments. And I gotta say, maybe because our model is a little bit more complicated than Gears, I gotta say maybe Gears can do much better than ours model in those lower quality data sets. But at least in high quality data sets that are more rigorously measured, our model performs better than Gears.
Thank you.
Yeah, fantastic work. I think my question kind of relates to the previous question, but specifically I'm interested in the out of distribution, I guess, generalization to like novel cell lines. Have you, I guess, split your validation data in a way that could assess whether, for example, your model is able to generalize to a new cell line, maybe in the same data set or maybe in another data set?
Yeah, actually, because we follow the procedure that Gears established, that is first way, as we can see from the method from the problem setting, we see all the control cells at the same type as a xi here, and if you are including new cell lines, that basically means the control cells are of a totally different distributions, and that would definitely harm the the generalization of this model. And so I gotta say, still the whole model, including gears, including our models, it's still for the same cell type. Our models innovation is more like it's only on the AI, on the AI predicting the post perturbation shifts. And for more generalizable models in the future, I think it's better, as I said, in this related works in recent years, and also introduced by the Dr. David. Yeah, very good presentation, by the way. We more likely will use pre-training models on very large scale database, on label database. And we utilize gears or our model as the downstream head for this more cell types. Yeah.
Thank you.